Well, good afternoon. I'm Greg Gray from Infectious Diseases. And if you want to get CME credit for this, please text BEMCAT to 919-213-8033. Today, we're going to talk about emerging respiratory virus threats, and I'll cover a number of these threats, not only the pandemic threat. First, my fiscal or financial disclosures are shown here. I have funds from a number of government agencies and diagnostic companies. Today, we're going to talk about some of the respiratory virus family groups that I think are most important, chiefly the top 5 there, as there have been new revelations, if you will, regarding these viruses that I think are important for people to consider. One of the key elements I think that we need to think about in looking at these respiratory viruses are that many of these are zoonotic. You certainly know that influenza and coronaviruses are zoonotic, but did you know that some of these other respiratory viruses have at least some viruses in their groups that have evidence of being zoonotic, and that meaning going either way, crossing species from animals to man or from man to animals or between other species. You might ask, well, which of these are the most important? Which ones should we invest in anticipating threats? Well, Dr. Emily Bailey from our group recently conducted a review with others of recent respiratory virus epidemics in the last 2 decades and looked at their relative impact and their association with animals, and chiefly focused on 4 different viral groups shown here: adenoviruses, enteroviruses, coronaviruses, and the various influenza viruses. And you can get an idea of the relative impact on man by looking at the size of the rings shown here. Perhaps a better illustration is this table that we've included in a recent submission to for NIH funding, showing that in the last 25 years, we've had really large outbreaks of enterovirus 71, causing an estimated mortality specifically in China or in Asia. Then, of course, in 2003, the SARS coronavirus affecting 29 countries, some 8,000 cases. But do you remember the killer cold virus, adenovirus 14? It's now affected people in at least 4 countries, caused a number of serious illnesses and deaths. Of course, in 2019, we had the influenza A pandemic that went worldwide, tremendous morbidity and mortality. MERS coronavirus in 2012, 27 countries now, more than 2,500 people infected. And maybe you don't know, but enterovirus 68 has caused quite a bit of morbidity throughout the world, particularly in the United States. And then finally, what we're faced with today, SARS coronavirus 2, which as of today's count has affected more than 175,000 people with confirmed infections. So these respiratory pathogens periodically strike us unawares and are something to reflect upon. How do we anticipate them, and how do we prepare, given that we know some are more of a threat than others? Well, some years ago, we collaborated with our veterinary colleagues in looking at 4 of these different viral family groups: the coronaviruses, the adenoviruses, the various influenza viruses, and the enteroviruses. And we asked our partners in the veterinary medicine, well, how do you detect new viruses in the animals that you seek to take care of? And basically, they and others have shared with us pan-species diagnostics, meaning diagnostics where we could pick up any human or animal virus. And we've attempted to build these algorithms focusing on specific known viruses in these groups, and then hitting the specimens that we collect in these various studies with pan-species diagnostics. And I'm pleased to say that this has yielded some interesting findings. Recently, we published a paper showing for the first time influenza D in poultry. We have some evidence of feline adenovirus in humans, vampire adenovirus from bats in humans, and most recently some canine coronaviruses in humans. So it just reminds me to say, if we don't look for these, the diagnostics we use in human or animal medicine are not going to pick up new viruses. You need to have a strategy to detect these, and we want to detect these early such that we can anticipate them becoming very, very effective in human-to-human transmission and virulent. Well, let's first go to the influenza A viruses. We've known quite a bit about these for a long time, since at least 1930s when our first detections were made. We know chiefly about influenza A and B, but do you know much about influenza C and D? These strains of viruses, these types of viruses have fewer gene segments, and they're also commonly seen in animals. In fact, influenza D is a virus that we've just recently learned more about, and it's being found in more and more animals just because we've only recently developed the diagnostics for it. Here you can see a nice graphic from a paper Dr. Bailey wrote from our group, showing that centric to all four of these influenza virus types are man and pigs. And so we often focus on the human-pig interface because we see that for various reasons, we are susceptible, like pigs, to the influenza viruses and probably to a number of other respiratory viruses. With respect to influenza A, you may have heard about the new hemagglutinin and neuraminidase types that have been found in bats, two different hemagglutinin and two different neuraminidase types. And up until recently, we didn't really think those were a threat to man. But one of the new developments has been that it turns out that they may infect you as well. So we need to be doing surveillance in bats to see what kind of influenzas are circulating, such that we can understand what would be a new threat to man from these very novel glycoproteins on the surface of these viruses. So a team in Zurich have shown that there's an alternative way for these viruses to infect man. So the ecology of the influenza A viruses have changed markedly since I went to medical school. I'm happy to report that we introduced the facts that put this camel on the map, showing horse viruses can move in camels. But there are plenty of other things that we've recently discovered here that shows that the ecology is much more complicated than we had anticipated 20 or 30 years ago. In fact, to do an effective surveillance for these, you really need to consider all the different animal groups that could contribute a novel virus to epidemics in man. Well, which of these viruses that are circulating would you focus on? I mean, and there are hundreds of these, you know, lots of unique strains that are immunologically important. Well, the CDC has come up with something they call an influenza risk assessment, and it's a quantitative and qualitative metric, expert opinion sometimes, where they evaluate the impact and the emergence of various different viruses that are circulating. And you can see here that the influenza A, H7N9 strains have been picked out as public enemies number 1 and 2, with a swine influenza H3N2 variant, number C here, as a close contender. And so the CDC is saying that's where we should put our investments in vaccine constructs, our phase 1 trials, our diagnostics. And it's a useful tool. With respect to H7N9, up until recently, this was the number 1 bad guy. It was circulating rather ubiquitously in China, causing periodic waves of infections in humans. In fact, the 5th wave was the highest we'd ever seen. And after that 5th wave, in about 2017, the Chinese government employed, as only China could do, a bivalent inactivated vaccine among all their poultry, free of charge. And you can see here that it markedly reduced the transmission of the virus in humans. It knocked down the carriage in the poultry and it reduced that transmission. And that's quite fortuitous because the virus was growing more virulent, becoming highly pathogenic in the birds. And so they basically saved a lot of their poultry industry as well as human infections. Now, will the vaccine hold? We don't know. There's always a chance that the vaccine could escape the vaccine interventions that are being applied in poultry, and we'll be faced with this threat again. But so far, it looks like it's working pretty well. And also, fortunately, we have not seen these viruses move into adjacent countries, not in large numbers anyway. And we're watching that very closely in partnership here at Duke with our colleagues in Hanoi. We're up on the border near China looking for incursions of that virus that they may see in their live bird markets. But if you ask me what I'm really concerned about as far as threats, it's all the viruses that are circulating in pigs. These viruses, for multiple reasons, have proven themselves to be a constant threat to man. And we know even less about the swine viruses than we do about the avian viruses because they're not a big problem in the swine industry. So they have some surveillance. It's usually clinically related and some vaccine interventions, but they're not as compulsive as we are, certainly in the United States, about ridding the poultry from all influenza viruses. And so the viruses are tolerated here, and they're growing in variety and they're growing in number. And yet we don't know what's going on, despite having a recent 2009 H1N pandemic that likely originated in pigs. So we asked the question recently in this review article in the International Journal of Infectious Disease, which of the viruses circulating in domestic animals are more likely to cause infections? And we compared the data between viruses circulating in dogs, cats, poultry, horses, and pigs. And in our opinion, the lowest threshold for infection for moving from the animals to man are the viruses that circulate in pigs. And because we know they contributed, or at least were associated with 4 previous pandemics, that's a concern. We also know that surveillance is very sparse, but the surveillance that is being conducted shows that the variants of viruses circulating in China, in the EU, and the United States are growing. There are many, many viruses, particularly after the 2009 pandemic viruses entered the pigs, many different variants circulating, often multiple variants on a single farm. So the opportunity for these viruses to mix and cause a new virus that causes a pandemic is a real threat, and one I would say of higher probability. So we have been talking quite a bit about how do we work on this threat? How do we find ways to work with the industry such that our research is not a threat to their animal production? And we've made the claim that unless we find ways to partner with them, ways to bring our technology to help solve their problems in a way that won't hurt their business, we're not likely to protect ourselves against these threats. So it's something to think about, particularly if you think that there might be an intermediate host in a domestic animal associated with the SARS coronavirus 2 infection. Well, let's mention those coronaviruses. We certainly have known quite a bit about these. We've had seasonal coronaviruses for many years, 4 different types. And then the emergent coronaviruses you've heard at length about here recently, SARS and MERS, that have emerged recently in various parts of the world. The SARS virus is thought to have risen in Guangdong province, that's southern China, in 2003. It affected a lot of people, some 8,000 people, 774 deaths. Fortunately, we were largely spared here in the United States and much of North America. and the virus has ceased transmitting with just a short period of transmission. Where it went, we don't know, but that's the good news. In contrast, the MERS coronavirus emerged in Saudi Arabia in 2014, and it continues to circulate, causing cases almost every week, particularly in Saudi Arabia, but also in the Middle East. This virus has a intermediate reservoir like the SARS virus, the civet cat and the SARS virus, and probably other animals. This one has an intermediate host in camels. So there's a lot of discussion about what can we do to mitigate infections in camels to protect humans. In fact, in monitoring the virus in camels, we've learned quite a bit that these viruses are changing. So we need to keep this in mind. The viruses that are circulating in our current pandemic may very well change. We need to keep this in mind because it affects our diagnostics, it affects our interventions, our vaccine constructs. And we hope that the viruses will change and become attenuated and cause less morbidity, but it's possible that they could become more virulent. So constantly evaluating the viruses as they circulate. Well, just like the Chinese have seemingly controlled H7N9 influenza A virus infections by administering an intervention to the reservoir animals, in this case poultry, there's been a lot of talk about administering an effective vaccine against MERS and dromedary camels. And we'll see, that may be the intervention, if we can find funding for it, that'll really knock down these infections. Because we know that one of the chief risk factors is being exposed to camel and camel fluids excretions. Well, with that in mind, 6 different coronaviruses emerging to affect man over time. Did you also know that we've had quite a few emerging coronaviruses in animals? This is a slide from Linda Safe at Ohio State, who's one of the world's most accomplished coronavirus animal researchers, and she notes quite a few coronaviruses emerging in pigs. And these viruses might emerge in Asia and, in weeks later, get here through one way or another, and they've caused a lot of problems in the animal populations here. Most recent outbreak is something we've called the SADS, S-A-D-S coronavirus. This emerged in southern China in 2018, killed about 24,000 pigs, and fortunately no people. It seems to be, again, a bat origin virus, and we don't know where it went and if it's going to come back. But it shows you that you need to be concerned about the viruses that are emerging in the animals, because sometimes they can cause explosive outbreaks and cross over to man. Well, what about the SARS-CoV-2 that causes COVID-19? It's basically now virtually everywhere we look for it. Some people might say, well, it seems to be less prevalent in many parts of the world. Well, I would say, if that's true, certainly it's influenced by poor testing. If that's true, it won't be for long. Most anywhere we go now, we could possibly be exposed to this virus. And it's explosively taking off in certain regions in the United States. So really, we're not safe anywhere. This virus could impact us, and hence the need for social distancing like we're seeing today that's unprecedented. Now, you might be asking, well, what do we know about virus spread? Well, we know traditionally these viruses spread by close contact, touching, coughing, and sneezing, and through inanimate objects, fomites, if you will. But there's some questions out there that bear some close scrutiny. There's been a recent paper to suggest that ocular infections could move through olfactory channels into the respiratory tract. That seems to be viable. There's some observational studies that suggest that fecal transmission may be a cause of human-to-human transmission, as perhaps was SARS. And the most controversial seems to be the aerosol transmission. How far is it safe to be in a room with a patient who is coughing, sneezing, or breathing viral particles into the air? Another question that's we're getting better focus on is how infectious is this virus? This basic reproductive number is often a measure of this. It's basically how many people are infected from a single case on average for a specific population. And you can see where we have various confined populations, such as the Diamond Princess cruise before they did any social distancing. That basic reproduction number was sky high, 14.8 cases per single case. Now you can imagine the situation in daycare centers, in schools, in prisons, where you have a lot of close contact, poor personal hygiene. That's going to be a similar high basic reproductive number. So we're trying to reduce that sort of social mixing. If you compare that with what they think is perhaps the overall mean of 2.7 cases per single case, you can see that even that is high compared to seasonal influenza and some of the pandemics. So this virus is pretty darn infectious compared to other influenza viruses that we've seen before. Also unique with this virus is it doesn't cause the typical upper respiratory tract infections we see with the other seasonal coronavirus. Instead, this causes fever and then it quickly moves into the lungs. So you get a dry cough, you get general fatigue, sputum, shortness of breath, and then you have chest x-ray and CAT scan findings. Sometimes you get gastrointestinal problems, but those are generally the more frequent signs and symptoms. The mean incubation time is anywhere 4 to 6 days. And the good news is about 80% of people will resolve this infection with little or no illness. It's people who are older, have immunocompromised airways, they're going to have the worst time. And when you look at the case fatality, that too is in somewhat of a contention. The case fatality is influenced by many different factors, healthcare certainly being one, crowding, likely being another, the intensity of viral load, exposure. At any rate, some people have estimated that the H1N1 pandemic case fatality rate is 0.01%, and SARS-CoV-2 is 100 to 400 times higher. If you make the assumption that for this virus, this viral pandemic to burn out, let's say it's got to affect about 70% of the population. We don't know if that's a good number or not. And you realize that in the United States alone, we have 327 million people. For us to see a cessation of this epidemic, we would have to see quite a few cases and deaths, 228,000 deaths. So it seems totally responsible, at least in my opinion, that we do what we can to reduce these fatalities as best we can and still try to maintain our way of lives. It's also being recognized now in this perhaps the largest review of the case fatality rates that the highest CFRs are in those that are the elderly. In fact, it goes way up when you're more than 80 years old. So we need to think about trying to protect them with the greatest interventions against for social distancing. Now, many people are still talking about containment, but I would argue that we have moved away from containment and now we are mitigating. We're trying to reduce transmission because the virus is essentially everywhere. It has not been contained. I argued this actually in February in an editorial in U.S. News and World Report, but we are now fully mitigating. And these mitigations, you might scratch your head and say, well, if Tony Fauci, the head of NIAID, says we're not going to have a vaccine for maybe 18 months, what does it matter that we prevent infection for some people over 6 months. Well, I would argue that it matters because over time, just like Ebola and SARS, we will get better and better at treating these things. We will know what supportive care really matters. Can we give positive oxygen pressure support? What sort of fluids? What sort of off-the-shelf antivirals do we have? Does interferon work? All these questions will be resolved, I think, or at least we'll have more data on them before we get a vaccine. So it's worth our spending the time to protect the people at highest risk. And the virus could also attenuate. Some people believe that may happen, and that would also reduce morbidity. So everybody's talking right now about flattening the curve. We don't want to be facing the situation that Italy is seeing, where their hospitals, and Wuhan has seen, their hospitals are overrun, and not with just the worried well, but with very sick people, such that they're having to make choices: who gets on a ventilator, who gets seen. We don't want to see that. We want to try to modulate, flatten the curve, if you will, these infections over time to save lives. Well, what are the interventions? I'll just say there's a whole bunch of these going on. In fact, China has registered, as of early March, more than 105 clinical trials. Now, some of these were some of the Chinese medicine interventions, and maybe of some question for those of us in allopathic medicine. But there are a lot of other things happening, some things that are consistent with our hypotheses regarding biological basis for interventions and our safety trials. One of the biggest ones I'm happy to say I'm engaged with is on the safety board. This is the Moderna mRNA vaccine that's already, I believe, in humans in a phase 1 trial. This is very exciting. It has a lot of promise. And there are multiple other vaccines on the horizon that are being examined. With respect to antivirals, remdesivir is thought to be the #1 best candidate. There's also another nucleoside inhibitor, EID 1931, that's thought to be good. And then some other medications were actually developed for other purposes, such as malaria treatment, you might know chloroquine, influenza, etc. So we'll see some of these will be eliminated quickly. Other compounds will be brought to clinical trials. One of the interesting things that we haven't seen in a long time is the idea to use plasma therapy. This is the idea that you take antibodies, plasma, from people who've recovered from illness, and you give it to people who are infected. Certainly this was used early on to fight other viral infections. We don't often see this, but there have been advocates of bringing this back just because we're likely to see so many deaths here in the United States. And here at Duke, a number of people are working on monoclonal antibodies that might be used therapeutically as well. So there's hope for new interventions. But there's also some cautions. This is a slide from Ralph Baric over at UNC, and he, in this slide, is summarizing some of the vaccine complications they saw for vaccines that were targeted for the SARS coronavirus, the first one. And so there are a number of problems to be overcome, I'll just say, both with respect to the immune protection and the safety of the vaccines before we can say we have something. And even if we have a vaccine that's very safe and effective, how are we going to pay for it? And how are we going to, Bill Gates says half the problem with vaccine interventions is getting the vaccine out to the world. It's a huge part of the equation. How do you move vaccine throughout the world to really knock down this pandemic? So tough times here, and we're making some tough social distancing calls. Let me share with you some of the interventions or some of this research that we're doing here at Duke in infectious diseases and our Duke One Health group. This is an epidemiological study that we're putting through the IRB right now, where we would study patients suspected of having SARS-CoV-2 and their close contacts. We'd be looking at collecting nasal swabs, rectal swabs, serum, acute and convalescent specimens, and doing bioaerosol sampling. This is run by our laboratory manager and postdoc or research scientist, Raquel Binder. In this study, we would set up very exquisitely effective samplers in regular wards. We don't want the negative pressure rooms because it won't work. In regular wards for patients who are admitted for care. And through this sampling, we can measure at 1, 2, and 3 meters out from the head of the bed and measure not only nucleic acid, but also we think viable virus in those specimens. So we can get at one of the gaps in knowledge, how far are people, when they even breathe, sharing the virus through small particle aerosol. These samplers were produced by NIOSH. They capture particle sizes in 3 different increments, and we've used them in about 10 publications in multiple settings, airports, subways in Singapore, all various different animal settings, and they're very good at picking up virus. Ben Anderson from our group is doing a challenge study of a number of different cell lines that we've archived over the years, epithelial cell lines in animals. And you may have seen the report in Hong Kong about a dog from a patient having a prolonged infection of the SARS-CoV-2 virus. Well, we want to understand if that dog or that cat or pigs even could be an intermediate host for transferring that virus to man. Maybe the dog is infected by a patient and it infects a person who doesn't have contact with that patient. Or maybe the dog even amplifies, increases, has a high viremia, increases the risk of transmission. We don't know, but we want to study that. And our first step is looking at if the virus replicate rather rapidly in these animal cell lines. So Dr. Anderson will be doing that here in the Biosafety Level 3 facility in our regional biosecurity lab. Leishan Zhu, a PhD student from China in our group, has developed a pan-species conventional RT-PCR coronavirus assay. This will be a very effective tool to go to the human and animal interface and not only detect the pandemic virus, but all known previous and new coronaviruses that may come from 4 different genome groups of coronaviruses. So we're very excited about this. It's simple, conventional RT-PCR with sequencing, and we've replicated the challenge studies here in our lab in Singapore, and it seems to be working really good with good sensitivity and specificity. So a new tool to discover new viruses that may emerge from the coronavirus family at the human-animal interface. Another group of viruses that we're concerned about are the enteroviruses. These are small viruses. There's a whole bunch of these different groups of viruses you've heard about. Certainly the rhinoviruses and polioviruses and enteroviruses. Enterovirus 68 has caused a lot of morbidity in the United States since 2014. We've had it in 49 states. It's caused periodic epidemics with significant history of asthma exposure. And most recently, it has been implicated as a cause of acute flaccid myelitis. And so we see these periodic outbreaks, don't fully know why, but they come and go, and they often affect children. Now we're getting more and more sophisticated in discovering the etiology of some of these rare, rather cryptic events. This is a nice paper that was just recently published in Nature Medicine, where colleagues use something called ViroScan or VirScan to interrogate some 481,000 peptides serologically with patients' CSF. And so the idea, is there evidence of antibodies in the CSF showing that specific viral groups might be a cause of the acute flaccid myelitis? And this sort of amazing non-bias, non-focus molecular cutting-edge diagnostics is where we need to go in the future, and it has proven successful in the etiology of this disease. This comes from a group, Joe DeRisi at UCSF, his technology there. Another enterovirus has caused, as I mentioned earlier, big problems in Asia is enterovirus A71, huge outbreaks. I remember going through a rural hospital in the jungles of Borneo and seeing posters of this, hand, foot, and mouth, the rash that's typical. This thing has really scared a lot of people because it's killed a lot of people. You can see the massive epidemic shown here, 12 million people affected in China, 2008 to 2015. And while we're seeing some outbreaks here, we're certainly not seeing the prevalence of virus that they're seeing in Asia. And so it has been in Asia where they focus a lot of interest in developing vaccines against this virus and all its different clades. And I'm pleased to say that it looks pretty good. There's a nice candidate vaccine. It seems to be pretty safe, at least in this early phase 1 report. So maybe Asia will develop and provide support for a vaccine that could reduce this morbidity. What's interesting to me is when they reclassified genomically a lot of these different various respiratory virus and other virus groups, a lot of the human enteroviruses were aggregated with animal enteroviruses here in the bottom in that big circle. And so there's a phylogenetic proximity of these viruses. And one wonders, well, what evidence do we have that these viruses might be crossing species? One of the groups in these viruses is called the rhinoviruses. And when I went to medical school, these were thought to be pretty innocuous, cause colds and weren't really a problem. We knew there was a type A and B, but since then, one of the new findings is relatively new that these viruses cause pneumonia. Sometimes it's a co-infection event with another pathogen. Sometimes it's the only etiology. We see this both in humans and adults. And so rhinoviruses suddenly have become important in our etiology of pneumonia. These were two very large CDC-sponsored prospective studies. We see periodic outbreaks of this, often in young children, hospitalization with rhinoviruses, sometimes in adults. The other new finding, relatively new, is that a whole new group of these were found, basically using molecular diagnostics, the rhinovirus C, and it turns out that these are somewhat virulent and causing a source of outbreaks we didn't know before, and something that we need to better understand and have developed some new diagnostics against. So some people in our group asked the question, well, what evidence is there in the literature that the enteroviruses are causing zoonotic infections? And I'll just summarize it by saying significant evidence. And we really ought to be looking more closely in our intensive workups of people with, for instance, pneumonia of unknown etiology for the enterovirus in some ways, because there's plenty of evidence of these moving back and forth between species. Let me move to human adenoviruses. We've known quite a bit about these since the 1950s. They're a big, stable, double-stranded DNA virus. There's now nearly 100 of these different types or serotypes identified. They cause a whole wide spectrum of clinical disease and tissue infections, and they're a big problem. We see huge outbreaks of these where people congregate, where they're mixing, such as military camps, people from different geographical areas bringing virus in, infecting people who are immunologically naive, and a lot of respiratory illness, some of it severe, in the military camps. So much so that they developed vaccines against these that were pretty darn effective. But recently, we've seen a rare type, serotype 14, emerge in the United States and cause havoc, and it continues to circulate today in multiple states and in multiple countries. We don't fully understand this, but this virus is not hugely different than the previously circulating prototypic viruses of that serotype, but it's caused a lot of outbreaks, and we continue to see it. Sometimes we see a lot of deaths. So this has been coined the killer cold virus. And because of this, and because of we're seeing these outbreaks, we have advocated that it's good to know the genotypes that are causing outbreaks in your hospitals. It's good to know for nosocomial purposes. Do you have a endemic virus in your intensive care unit, in your ward that treats patients who are immunocompromised due to organ transplant? And so we've played a role for that here. We can type these and help you get a handle on the viruses that may be circulating. And because we've seen some emergent strains, the adenovirus enteric-coated vaccines have been thought of as a possible model because these are so safe and effective for adenovirus 14. We'll see if those are ever produced if the adenovirus 14 comes back. We know these vaccines, they're given at the same time, have reduced 100-fold the disease burden in the military and prevented deaths every year, perhaps as many as 2,700 hospitalizations. So they're really thought to be a godsend, if you will, in reducing military camp morbidity from respiratory illness. But we're now seeing China, realizing that they've had big outbreaks, come up with a trivalent vaccine. This is inactivated, and it seems to be working at least immunologically in early trials. Instead of targeting 4 and 7, they're targeting 3, 7, and 55. Right now, we don't really have FDA-approved antivirals, and we'd only have vaccines against 4 and 7, but there are some promising data regarding an antiviral that was produced for smallpox and other, actually it seems to be effective against other DNA viruses, such as adenovirus. This antiviral is produced here locally in the Durham RTI area. Seems to be pretty darn good and safe, at least in initial trials, in treating severe adenovirus disease. Well, we asked the question, is there evidence that these viruses may be crossing species? We had noticed a number of case reports, particularly in non-human primates, where people working with non-human primates were being infected with adenoviruses that were not supposed to cross over. And even somebody working with a parrot colony was infected with an adenovirus in parrots. So our group did a systematic review again of evidence. And again, there was quite a bit out there that we didn't know in the literature that these viruses could cause epidemics or cause infections in man. And so again, we need to find ways to look at the human-animal nexus and anticipate adenovirus threats. The last virus group I want to mention are the human metapneumoviruses. This is a single-stranded paramyxovirus, pleomorphic and slow-growing, and we really didn't even know about it until 2001 when a group from the Netherlands accidentally discovered it. Since then, we've noticed that this virus is everywhere. It causes infections year-round. It causes infections in children and adults. Outbreaks can be nosocomial. You can have death. It can cause severe pneumonia. So this is often on the menu for the multiplexing assays. And we see outbreaks like this, particularly in long-term care facilities, causing somewhat severe disease. So human metapneumovirus is a contender for our attention. Now, what's interesting is what happens sometimes when you go to science meetings. I got on the human metapneumovirus bandwagon soon after it was discovered when I was at the University of Iowa. We had collected some 58 viral isolates and characterized them into 4 genotypes from patients in our hospital. And at a meeting for influenza, I met scientists who studied the closest related virus to the human metapneumovirus, the avian pneumovirus. Now, this virus plagued the turkey industry. It caused so much disruption in Minnesota that they moved all the turkeys indoors. Passerine birds, little birds that were moving the virus from turkey flock to turkey flock and wiping out all the flocks. So they've come up with interventions to protect the biosecurity. My colleagues at University of Minnesota, with whom I was meeting, had the freedom to do something we cannot do in human medicine, we don't want to do, and that is to challenge the animals they cared for with live human virus. So basically, they took the human metapneumovirus and they inoculated the birds and showed that all 4 genotypes cause signs of infection and pathology of infection and molecular evidence of infection. So here's a human virus that has potential to cross over to turkeys that we didn't even know about until we looked for it. In a similar fashion, we asked, can the turkey virus move over to humans? So we engaged graduate students in this kind of work. Ghazi Kayali, who's been on the Netflix epidemics thing recently, in the bottom here, and Ernesto Ortiz on the top, teamed up. Ernesto is here in DJHI, teamed up to study turkey workers in Iowa. And they drew their blood, and we developed a rather crude assay to look for evidence serologically of the turkey virus. And I'll just say that the turkey workers had pretty strong evidence of their immune systems being challenged with those antigens. Were they infected? We don't know, but it was much higher than controls. So again, unless you partner with people in the veterinary community, you won't learn about these cross-species infections unless you have tools to detect them. So let me just say that I've covered 5 different viral groups here. There are other important viruses, but there's not a lot changed in the last 10 years with respiratory syncytial parainfluenza and Boca viruses. We still don't have good interventions for those, and they still cause problems. A number of respiratory virus types are prone to cause epidemics. In summary, many of these originate in animals. We need to find ways to work with our partners in the veterinary world and in the animal industries where they grow large numbers of these animals, often for food purposes, to detect these before the viruses adapt to man. We want to do this in a One Health way because we know we're all interconnected, human, animal, and environment. And we want to catch these viruses before we want to catch them right here early on the left, when people are really essentially able to stop the infections. Our natural immune system blocks the infections, but we can still see if their immune system is challenged, because over time, the tendency is the virus can become more susceptible and adapt to man, and then become highly efficient in causing transmission, like we see today in SARS-CoV-2. So we need to catch it early on so we can better develop interventions and diagnostics. With that in mind, we've pitched a very ambitious study to the NIAID. I'm waiting to hear, but this would call for 2 years of surveillance in Asian of people working in the live animal market industry. In these studies, we would prospectively follow 400 people, 80 controls. We would sample them with nasal swabs and sera, and we would sample their animals and the feces of the animals, looking for viruses that might emerge. And we would use another tool by Joe DeRisi at UCSF. This is Star Wars-like. It's basically a way to do big data analysis that's not focused on specific viruses, but to look in a categorical way, so that in a way that you can quantify everything, to look for virus groups that are in specimens. So we've used this recently in fecal slurry here in North Carolina and found a lot of viruses we didn't know about, and also used it in aerosol sampling. So non-biased metagenomic sampling, where we would compare the viruses that are circulating in the animals to the viruses circulating in the nasal passageways. And if we see a new virus has gone to both, then serologically we could evaluate the patient's sera, look for evidence of infection. So this is how we would do a big data reduction strategy to look for the next emerging threat. So thank you very much. This is a graphic of our work, One Health research team. We're about 30 people, 30 pathogens, focused on 30 pathogens, 30 projects in 13 countries, with labs here at Duke, Duke NUS, and Duke Kunshan. Thank you. I'll take questions.
Hi, everyone. This is Libby. You can type questions into the
I have one like I'm quite interested as much as you're in the COVID and stuff like that. What about rhinovirus? You talked about it, A, B, et cetera, et cetera. It seems like up on our transplant population, there's an awful lot of rhinovirus up there. And it also persists long periods. So have we got any concreteness to really look at that class virus a little bit more, particularly?
We could, yeah, so the question is, what is rhinovirus playing a role and on some of our immunocompromised patients here at Duke, and could we look at it more comprehensively and get a handle, I guess, on the viral genotypes and specificity of, you know, maybe association? I think we could. We would have to, you know, develop. We don't have a pan-species rhinovirus assay right now, so we would have to develop a new diagnostic for that purpose. But that's something we could explore. And what's surprising is those large prospective studies the CDC sanctioned, the rhinovirus was a huge player, unexpectedly. People don't understand that at all. So, you know, is it a cofactor? We don't know. It's a good thing to think about. All right, well now, if you wanted to receive credit for this, let's go, let's go back to that slide and say, you need to type your CME code here to that phone number in a text message. Any other announcements? We're gonna archive this somewhere. Where would we archive it? Okay. Well, thanks everybody in the room who attended and for your kind attention, and those of you who listened in through the webcast. Bye-bye.